This is a diagram depicting the 5 ways a molecule can pass through a cell membrane. The first 2 ways a molecule can pass through a cell membrane are both forms of diffusion. The first form of diffusion is called simple diffusion. This process is a form of passive transportation, meaning it requires no added energy for it to occur. The molecules that are able to move through are nonpolar because they will not get stuck to the hydrophilic head of the phospholipids in the phospholipid bilayer. There is no transfer protein involved. As you can see, this process goes with the gradient, which means the molecules diffuse from high concentrations to low concentrations. The molecules passing through are all small monomers. The second form of diffusion is called facilitated diffusion. This is different from simple diffusion because through the use of a transfer protein, polar molecules and ions can pass through the cell membrane through the canal-shaped opening in the transfer protein. This process is also a form of passive transportation, meaning it requires no added energy for it to occur. As you can see, this process also goes with the gradient. Although not shown here, the molecules passing through can vary from small to larger size monomers. The third way molecules are able to pass through a cell membrane is called active transport. This is different from diffusion because it requires energy in order for it to occur. It still uses a transfer protein, but any ionomer can pass through the membrane through the canal-shaped opening in the transfer protein. As you can see, unlike the previous processes, this form of transportation goes against the gradient, which means molecules travel from low to high concentrations. This is not a natural process, so it requires the use of additional energy. The last 2 ways a molecule can be transported across the cell membrane is through vesicular transportation. Vesicles facilitate the transport of specific molecules across the cell membrane. The structure of a vesicule is a phospholipid bilayer with a macromolecule enclosed inside, similar to a cell membrane. When a macromolecule is excreted out of a cell through vesicular transportation, it is referred to as exocytosis. When a macromolecule enters a cell through vesicular transportation, it is referred to as endocytosis.